Hello, everyone. Welcome back. Lecture 15 in Virology 2020, Mechanisms of Pathogenesis. We've talked about immune responses the last two lectures, so now it's time to go back and look at the ways that viruses cause disease, of course, which is pathogenesis. And I don't know if you've noticed in all of these lectures, I have a little quotation on the front left of the first slide. This is perfect. We have met the enemy and he is us. We'll get into that today. First, let's talk a little bit about how we study pathogenesis, what the goals are. And a key part of pathogenesis is, of course, having an animal model. You can only do so much in humans. You can do observations, of course, when you have an outbreak, and that's good. And for some viruses, you can actually infect volunteers. So rhinoviruses, which cause mild infections, you can actually infect people. And there have been some studies done where they put people around a table in a room playing cards. And they have one person who has a rhinovirus infection, and they watch it spread to the others via the cards. You can do it with influenza viruses. You can do it with noroviruses. But for the most part, we cannot do human challenge. It's not ethical. So we use animal models. And we always have to remember it is a model. It does not predict what's going to happen in humans. You get observations in mice, and you have a lot of things you can do that you can't do in humans. You can make genetically altered mice, transgenic mice. But you never know what you find is the same as what will happen in humans until you... In some cases, you can do some experiments to validate what you find in the mouse model. And we always say mice lie and monkeys exaggerate, which is a broad way of saying don't consider that any animal model predicts what happens in humans. So even non-human primates, which are very close to us, 99% identical genomes to humans, they can have very different outcomes with the same virus infection. And here's a slide just that illustrates what can be done with a mouse model. So you can study virus infection in disease in mice, and you can use laboratory mice. You can modify them in various ways to help what it is you're trying to study. You can introduce clonal T cells, receptors. You can delete genes encoding immune mediators. You can delete immune cell populations themselves. You can overproduce immune mediators, right? So all manipulations of the immune system. Then you can make transgenic mice that produce human receptors for viruses. Sometimes we put the entire viral genome in a mouse for hepatitis B virus. That's been instructive. Or you can produce individual viral gene products in mice. You can also modify mice to do what we call humanize them. We can put human transplants, which would be xenografts, different human organs, and put them in mice that will repopulate the mouse. And you can make a mouse model for HIV infection, for example, which infects the immune system. So you put components of the human immune system into mice. So then you can infect the mice. You can see virus replication, where it is and what levels and so forth, and you can study immune responses. Now, in general, when we use animal models, we have two broad approaches that we use. In one case, we take human viruses and infect animals with them. So you can find a virus that will reproduce in a mouse, either a wild-type lab mouse or a mouse that you've modified in some way to make it become infectable. But not all human viruses infect mice, in which case sometimes we work on animal viruses that resemble human infections. We find viruses that naturally infect mice, for example, that are related to viruses in humans, and we can use those to make observations and see if they parallel what happens in humans. Let me give you an example of a mouse model that we actually made in our laboratory in the 1990s. And this started with the identification of the poliovirus receptor in my lab by Kathy Mendelson, who was a graduate student at the time. We then used the human gene encoding the poliovirus receptor to make a poliovirus receptor transgenic mouse. And that was done by another graduate student in my lab, Rebao Ren. And then we infect this mouse with poliovirus, and the mice become paralyzed. As you can see, hind limb paralysis here. So mice are permissive to poliovirus infection, but they're not susceptible. They don't have a receptor. If you infect mice with most strains of poliovirus, the virus will not replicate because it can't get into mouse cells. However, all you need to do is put the receptor into the mouse germline as a transgene, and now all the many mouse tissues produce the poliovirus receptor, and you can then inject them with polio and they become paralyzed. So this is a great model for studying where the virus replicates, how it causes paralysis, and so forth. And other mouse models for viral diseases have been made since this. Another topic I wanted to touch on is tropism, tissue tropism, the spectrum of tissues that are infected by a virus. In general, when viruses infect animal hosts, they don't infect every tissue and every cell type. They have a restricted tropism. They can be, for example, enterotropic, which means they replicate in cells of the enteric tract, neurotropic, neural cells, hepatotropic, liver cells, and they can be strictly hepatotropic, or they could be a combination of hepatotropic and enterotropic, for example. But there's always a pattern of tissues that are infected. The tropism can range from limited, so we could have a strictly neurotropic virus, to pantropic, where the virus infects many, many different tissues. And one of the goals of studying viruses is to understand what are the determinants of tissue tropism. And that way we can understand what restricts viruses and maybe use that in some way, in some useful therapeutic way. And some of the determinants that we've learned over the years that control the tropism of a virus include susceptibility, whether there's a receptor for the virus on the cell surface. So mice are not susceptible to poliovirus. No tissue in a mouse can be infected with poliovirus because there's no receptor. So in a way, tissue tropism and host range are very similar properties in that similar determinants are involved. Mice are not susceptible for poliovirus, but if you put the receptor in, as I just showed you, they become susceptible. They're already permissive, apparently, because the virus gets in those mice and there's nothing else but the polio receptor and everything else is mouse, and the virus will replicate. So permissivity, though, for many viruses is a determinant of tropism. Some viruses can enter a variety of cells and don't replicate in there because some crucial protein is lacking. Accessibility of cells. Some cells are simply physically inaccessible to virus and won't be infected. And immune defenses of all sorts, ranging from intrinsic through to adaptive, can regulate the tropism of viruses. Let's look at some examples of this. A really nice one that I like to cite is what controls the tropism of influenza virus. So influenza viruses, of course, human influenza viruses are respiratory pathogens. They typically replicate in the upper, middle, and lower respiratory tract. They cause respiratory disease, and typically they do not replicate elsewhere. And we think that cleavage of the viral hemagglutinin is an important determinant of that restricted tropism. So I remind you that the viral particles, envelope influenza virus particles, have glycoproteins embedded in the membrane. One of them is the hemagglutinin, which is important for binding to receptors and catalyzing fusion in the endosome upon reduction of the endosomal pH. So here's a line diagram of the viral HA protein, just to remind you that there is a cleavage site just before the fusion peptide. That has to be cleaved in order for fusion to occur at low pH. And we think the proteases that cleave the HA here, that little orange arrow, that's the cleavage site. We think those are cellular proteases, and their availability in different tissues regulates tropism. And an example is a trypsin-like protease, shown on the lower left side slide here, made by specific cells in the respiratory epithelium called club cells. epithelial cells surrounding a club cell. This club cell makes this protease, just tryptase, which we think cleaves the HA of influenza viruses as they are released from the cells. And those viruses with cleaved hemagglutinins can then go on and infect other cells. Now, if these viruses should happen to get into, say, a liver cell or a kidney cell, they may be able to attach and enter those cells because sialic acid is ubiquitous. They may be able to infect those cells because the HA is cleaved. It was cleaved in the respiratory tract. But the viruses that are produced from those individual cells will not have a cleaved HA because there's no tryptase in those other tissues. And so that restricts the replication. So the idea here is that the proteases are restricted to the respiratory tract, and that's what keeps virus tropism restricted to the respiratory tract. There are some other proteases that are also involved in cleavage of HA, not just this one. This is a secreted protease. There's another one, a very important one, TMPRSS2, transmembrane protease serine 2. This is an endosomal transmembrane protease. And this has been shown to be important for cleavage of HA of human influenza virus strains. The idea here is that this protease would be present in the endosomes of these respiratory cells, and the viruses would be cleaved on the way into the cell. So they're cleaved in this case by tryptase on the way out, can also be cleaved on the way in, cleavage during entry. There are avian influenza viruses that have a different cleavage site. And these are highly pathogenic avian influenza viruses, H5N1, and we'll talk about those a bit later. These viruses cause outbreaks in birds, and occasionally they infect humans, typically humans who are in contact with birds, and they cause very serious disease, and viruses appear to replicate in many different tissues, not just the respiratory tract of humans. Sequencing of the viral genome reveals that they have a different cleavage site upstream of the fusion peptide. They have what's called a furin cleavage site. It's a multi-basic amino acid, a series of multi-basic amino acids. And that allows the HA to be cleaved by furins, which are proteases inside of the cell that are probably cleaving the HA on the virus particle as the HA is transported from the ER to the plasma membrane. But the key here is that furins are ubiquitous. They're in many, many different cells and tissues. And so that expands the tropism of these avian H5N1 influenza viruses. So cleavage of the HA of influenza viruses is a determinant of tropism based on the availability of the protease, the cell protease that's cleaving that. And yes, these cell proteases are targets for antiviral interventions. That's being pursued in a number of places. This brings us to the current coronavirus, SARS-CoV-2. So let's talk about cleavage of the spike, very much like cleavage of the HA, and how it regulates tropism and host range of coronaviruses. So here's our coronavirus particle to remind you. It's an enveloped virus with spike glycoproteins embedded in the viral membrane. This spike mediates attachment to cell receptors. The viruses enter by endocytosis, and upon acidification of the endosome, the viral and cellular membranes fuse and the RNA is deposited in the cytoplasm. The low pH dependent fusion requires cleavage of the spike, very much like cleavage of the influenza HA. And for many coronaviruses, it is known that endosomal proteases are responsible for the cleavage. So the cleavage happens on the way in. Cathepsin is a set of endosomal proteases that can cleave the spike of coronaviruses. Again, TMPRSS2, should be a 2 there. Endosomal transmembrane proteases can cleave the spike on the way in. That's been shown not only for SARS-CoV, but SARS-CoV-2, the currently circulating strain. That TMPRSS is one of the proteases that can cleave it. However, there's another interesting observation which I want to tell you about, and that is when this genome sequence of SARS-CoV-2 was done back in January, the sequence of the spike revealed a furin cleavage site, which is not present in other coronaviruses. There is the diagram of the spike amino acid sequence. And here's the structure of the spike, which was solved not too long ago, an amazing feat of very rapid structure determination. And this spike glycoprotein structure, which corresponds to the spike is a trimer, so this is a monomer. Here we have the receptor binding domain of the spike. It's a separate part of the protein, and that binds the receptor ACE2, which is the receptor for both SARS-1 and 2. And then we have two cleavage sites for proteases. There's one here, S1, S2, and a second one, S2. And there's your fusion peptide in the light blue, and you can see that colored in this structure. The fusion peptide is buried down there. So cleavage is needed to allow low pH dependent exposure of the fusion peptide during virus entry. Interestingly, now we talked about this before, the SARS-CoV-2 is extremely similar in genome sequence to a bat virus called RATG13, which was isolated in 2013 in a cave in China about 1,000 miles outside of Wuhan. It's a bat SARS-like coronavirus. Probably shares an ancestor with SARS-CoV-2. So SARS-CoV-2 is clearly a bat virus. If you compare the genome sequence of the human SARS-CoV-2 and this bat virus, you can see that there is a furin insertion sequence here, right between S1 and S2, and here on the structure diagram, which is not present in the bat virus. And it could very well be that that furin sequence is cleaved and helps to activate the spike for fusion. Those experiments, I'm sure, are being done in many laboratories right now. So again, furins are ubiquitously expressed, so the presence of a furin cleavage site would allow the virus to replicate in many different tissues. And how the virus acquired that, the bat virus does not have the furin cleavage site. It probably was introduced by recombination just randomly with another coronavirus, and that probably helps it to replicate in human cells, as well as the changes in the receptor binding domain that allow it to bind human ACE2. There are a number of changes there with respect to the bat coronavirus that likely are responsible for a high affinity binding to human ACE2. So anyway, two examples where proteases can regulate the tropism in the host range of viruses. Our first question is, insertion of multiple basic amino acids at the HA cleavage site allows influenza virus to infect many organs. This means that the blank of the virus has changed. So blank is susceptibility, club cell tryptase, permissivity, tropism, all of the above. The answer is the tropism of the virus has changed. The highly pathogenic avian influenza viruses have multiple basic amino acids at the HA cleavage site, and that allows cleavage by furins, which give it a broader host range. Okay, now let's talk about viral virulence, the capacity of a virus to cause disease in a host. An important concept and important to understand how it's measured. We talk about virulent viruses that cause severe disease, and we talk about avirulent viruses that don't cause any disease. And sometimes another word we use is attenuated to describe a virus whose virulence has been substantially reduced. For example, attenuated vaccines. These are infectious vaccines that have reduced virulence by some modification of the genome, and we will talk about that in our vaccine lecture. We can quantitate virulence in the laboratory. It's harder in people, and there's a very rough quantitation, which we've discussed before, the case fatality ratio, which, as I said, is very difficult to interpret because you have to know all the people who are infected to know that ratio. But in a laboratory, we can be highly quantitative. We can measure virulence in a number of ways. For example, We can simply measure virus titer in a host. We can measure the mean time to death, the mean time to appearance of signs. And remember, what you can feel is a symptom. So a mouse may have symptoms, but we don't know it because we can't talk to the mouse. So this would be signs in animal models. Things that we can detect. We can measure fever, weight loss, pathological lesions. Typically for polio vaccine testing, we measure the lesions caused by the virus in the central nervous system. Or an example with HIV-1, the reduction in the number of blood CD4 positive lymphocytes. These are a consequence of virus replication and the immune response, as we'll see later. Just remember that. Here's an example of measuring viral virulence, two different groups of viruses. On the left, experiment done in my lab many years ago, we are injecting mice with polioviruses. And these are, on the y-axis, the number of survivors. So this is a typical way of plotting virulence data by simply how many animals are surviving with time post-infection on the x-axis. And you can see we have two different serotypes of poliovirus, type 1 and 2. Type 1, all the mice survive after intracerebral inoculation. Type 2, eventually they all die by day 10 or 11 or so. And a dot at each day indicates the death of one or more animals. So measurement of survival. On the right, a different kind of virulence measurement. We are looking at what we call a neurovirulence score. So here we've used a variety of different viruses that cause CNS disease in mice. These mice are injected with the viruses, and then at times after infection, the central nervous system tissues are removed, and they're sectioned and looked at under a microscope, and a pathologist assesses how much damage they can see. And they assess a score to it, and that's called the neurovirulence score. And here we're looking at sections in the cerebrum, brainstem, and spinal cord, three different areas of the CNS caused by different viruses. And you can see some viruses cause a great deal of neurovirulence, a great deal of damage, and there can be differential damage in the CNS. So here, yellow fever, the vaccine strain, is causing more damage in the spinal cord. Same thing with West Nile. And this one, dengue, put right in the CNS, is minimally neurovirulent. So again, pathological lesions as a way of measuring virulence. You have to remember, however, that virulence of viruses is a relative property. It's influenced by dose, the route of infection, the kind of animal, the age, the sex, the susceptibility of the host, which means you can't compare the virulence of different viruses. For similar viruses, you have to use the same assay. So if you want to compare virulence of polioviruses, different serotypes, that's fine. You'd have to use the exact same assay in the same animal. Otherwise, you can't compare them. And that for the same reason, you can't compare the virulence of poliovirus and Ebola virus. And this is also why using case fatality ratios in humans is a poor way of measuring virus virulence, because the population, when you compare China with the U.S., different populations, not only different genetics, but different environments and different healthcare systems. So you really cannot do that. And that's why in the laboratory we keep the assays the same and we're able to compare virulence in those ways. Here's an example of how important it is to keep the experiment the same. Virulence depends on the route of inoculation. Here is a virus that infects animals, is used as a model for infection in the laboratory. Lymphocytic choriomeningitis virus, an enveloped rhinovirus. Here we are putting 100,000 PFUs intraperitoneally into a mouse. They all survive. No problem. We put one PFU intracranially, all the mice die. The simple change of the inoculation route makes a huge amount of difference. And for those of you thinking about how many viruses of SARS-CoV-2 you need to start a respiratory infection, well, we can't do that experiment in people. At some point, it'll be done in an animal model. But here, for this one, in mice, it's one PFU when the virus is put right in the brain. Route of inoculation is important. Our next question is, which statement about viral virulence is wrong? A, it can be influenced by dose, route of infection, species, age, sex, susceptibility of host. B, it can be quantitated by measurement of fever. C, Ebola virus is more virulent than human papillomavirus. D, it's the capacity of a virus to cause disease in a host. E, when comparing virulence, the assays must be the same. The wrong answer is Ebola virus is more virulent than human papillomavirus. You can't compare virulence of different viruses. Can't do it. Viruses are too different from each other. In virology, many people study viral virulence to identify the viral and the host genes that determine it. What genes make a virus virulent as opposed to less virulent, and what cell genes do the same thing. And we typically do this by mutation. The ultimate experiment. We introduce mutations into viral genes, and we look for viruses that cause reduced or no disease in a system in our animal model. And then we can study the function of those gene products. Let me give you an example of this. Here's some experiments using mice as an animal model to identify viral virulence genes. And we have a virus, a wild-type virus, which grows very nicely in cell culture, makes nice plaques. We can inoculate it intracerebrally into mice. The virus will reproduce, and it will cause disease. It will cause disease of the central nervous system. So we say the virulence phenotype is neurovirulence. This virus is neurovirulent. Now, let's say we introduce a mutation into this viral genome, and that mutation produces a general defect in reproduction. The virus reproduces poorly, lower titers, smaller plaques. When you put that virus into mice, it doesn't reproduce very well, and we say it's attenuated. However, I would argue that this kind of mutation isn't particularly interesting because the mutation is simply reducing viral replication. What you want is a mutation in a gene specifically required for virulence, but not for replication in cell culture. So here's another virus where a mutation in a gene doesn't affect reproduction in cell culture. When you put it intracerebrally into mice, the phenotype is attenuation. So the virus does not reproduce well in mice. So this is a gene that has something specifically to do with mice, and then you can take it and do experiments to identify what the function is. That's a virulence gene as opposed to a gene just generally required for reproduction. Which is not to say that such genes generally required for reproduction aren't interesting, but if you want to study virulence, you want genes specifically involved in regulating virulence. Now, these experiments have been done for a number of years in many different viral systems, and many viral virulence genes have been identified. And these genes, as you might guess, they influence viral replication cycles, but they also can affect invasiveness in tissues. So a virus put peripherally will not invade the central nervous system because of a certain mutation. That's quite interesting. They can affect the tropism. These are genes that modify host defense mechanisms. Some of the viral immune antagonism genes that we've been talking about previously are virulence determinants, because if you take away the ability of the viral gene product to antagonize immune defenses by changing the sequence of the protein, you're reducing virulence. We have other genes that enable virus to spread in a host. We have other genes with intrinsic cell-killing effects. So let's look at a couple of examples of these. First, a virulence gene does not actually have to be a gene. It could be in a non-coding region that doesn't encode any protein. An example of this, the Sabin vaccine strains of poliovirus. There are 3 Sabin vaccine strains, 1, 2, and 3 of poliovirus that are currently being used in the eradication effort. These contain mutations in the 5' non-coding region that reduce the neurovirulence of the virus when specifically introduced into animal models. So here's the poliovirus genome, 7.5 kilobase plus stranded RNA with a 5' untranslated region, and that region's expanded here at the left. It's highly structured, as we've discussed previously, and it contains an internal ribosome entry site. The 3 Sabin poliovirus vaccine strains all have single base changes in the 5' non-coding region, specifically in this stem loop 5, which is expanded on the right here. And these mutations are shown here in red for the type 1, the type 2, and the type 3 vaccine strains. Single base changes in each case from here G to A to G, and C to U. One base change in this 5' non-coding region reduces the virulence of the virus, and that's part of the reason why it's an attenuated vaccine strain. I'll show you an experiment to show that. This was done in my lab a number of years ago. Here we are measuring both the virulence of the virus, determined by lethality, and replication in the mouse brain. We have 2 viruses here, 2 polioviruses, which differ only at one base, at 472. So let's go back and I can show you. 472 is the base change in the type 3 vaccine strain, from a C in the virulent virus to a U in the vaccine strain. So now we have a pair of otherwise identical viruses, with one with a U and one with a C at that position. The virus with a C is lethal. 9,000 PFU will kill half of the mice after intracerebral inoculation. The virus with a U is completely attenuated, avirulent when it inoculated intracerebrally. So you can't reach an endpoint, no matter how much virus you put, and you're limited because of the titer of the virus. But even at 2 times 10 to the 7th PFU inoculated intracerebrally, no mice die. So that phenotype is paralleled by replication. Here's a time course days after infection, intracerebral inoculation of these 2 viruses into mice. And on the y-axis, we have plaque-forming units per gram of brain. And you can see the virus with a C, which is neurovirulent, replicates quite nicely in peaks at about 5 days post-inoculation, whereas the virus with a U, it doesn't replicate and is eventually cleared. So a single base change abolishes replication in the CNS and leads to attenuation of neurovirulence. Many gene products that modulate host defenses have been identified as virulence determinants. As I said before, this is obvious why this would be. The gene product antagonizes the immune response, allows the virus to replicate and spread and cause disease in the host. If you take that away, it impairs that ability because the immune response will clear the infection. And these immune modulators include proteins that regulate apoptosis, autophagy, and intrinsic proteins like the deaminase ApoBec3G, virokines and viroceptors. These are viral proteins that are homologs of chemokines and chemokine receptors. And so the viral homolog of the chemokine would bind the chemokine receptor but not have the same effect. So it would block the normal chemokine from binding. And in the same way, this viroceptors are soluble chemokine or cytokine receptors, which will bind the normal cytokine or chemokine produced by the host and prevent it from binding its receptor on the cell surface. There are also proteins that bind the complement pathway. There are modifiers of MHC1 and 2 pathways, which we've talked about. And all of these genes are typically not required for growth in cell culture, where we don't have these immune responses. So they fit the definition of a viral virulence gene specifically needed for the production of disease in an animal. Here's an example of an immune modulator that is a viral virulence gene. And this is a gene identified in the genome of a mouse herpes virus, gamma herpes virus 68. And this is a mouse virus that's used as a model to study or to understand human herpes viruses. The genome encodes a protein called M3, which encodes a chemokine receptor. And again, these are soluble proteins that bind chemokines and prevent the chemokines from binding their normal receptors on the cell surface. So you can remove the gene encoding the chemokine receptor, the M3 gene, and the virus will grow quite well in cell culture, and then you can inoculate animals. So here we've taken mice, inoculated them intracerebrally, and we're measuring virulence in this assay by the percent survival. So you take the wild-type virus in blue, and you add increasing doses of viruses intracerebrally, and you can see with increasing doses you get less survival. So at the left here, 100% survival. At the right, approaching 0. So that's the curve for blue, and you could calculate the 50% survival as about, 5 PFU or so. So that would be the LD50, lethal dose 50%. Now let's take away the M3 gene, the delta M3 that's green, and now you can see that it's less virulent. And the 50% lethal dose is now over 100 PFU compared to 5. So removing that chemokine receptor reduces the virulence of the virus. And then the third curve in red, we have added back the M3 gene to the mutant genome, delta M3, with marker rescue. So we put it back to make sure we haven't introduced any other mutations in the viral genome in all these manipulations, which are done, of course, by recombinant DNA manipulation. And that virus in red has now regained the virulence of the wild-type virus. So the gamma 68 M3 gene, which encodes a chemokine receptor, is a virulence determinant. A few viral genomes encode toxic viral proteins that are soluble. And this is an example of one called NSP4, encoded in the genome of rotaviruses. These are double-stranded RNA viruses that cause gastroenteritis in humans. And we think the NSP4 protein is what we call a viral enterotoxin because it is toxic in the enteric tract. So this protein, it's produced during virus replication in these intestinal epithelial cells. It inhibits the sodium glucose luminal transporter. That transporter is needed for water reabsorption in the intestine. And so water is not reabsorbed. So we think its inhibition by NSP4 could be one way of causing the diarrhea that's associated with infection. At the same time, the virus also induces a phospholipase C-dependent calcium signaling pathway, which results in the increase of intracellular calcium, and we think that induces calcium-dependent chloride secretion, which contributes to the failure to retain water. So we think this enterotoxin is a big player in the production of diarrhea caused by infection. So those are some examples of viral genes that regulate virulence. How about some cellular genes? Because, as I said earlier, the cell makes a big contribution to virulence, not just the virus. And so these kinds of studies have been going on in a number of laboratories. Let me tell you just two as examples. One is understanding cellular genes involved in herpes simplex encephalitis. So typically, we are all infected with herpes simplex viruses early in our lives. We have a primary infection in our mucosal surfaces, typically of our mouth. And we will talk about this more in 2 lectures when we talk about persistent infections. But these viruses cause a primary infection, which may or may not be associated with a cold sore. The virus spreads to local peripheral ganglia and becomes latent there, where the infection goes away in a few weeks. And then periodically, those genomes are reactivated, so you get another cold sore. And this is a benign set of circumstances here. Occasionally, on reactivation, however, instead of the virus going to the mucosal surface and making another cold sore, it goes into the CNS, and you have encephalitis, which can be deadly. This kind of spread can happen after the primary infection as well, where you might get a viremia and dissemination to other organs and eventual invasion of the CNS. So this is the fatal CNS infections caused by herpes simplex viruses occur at a rate of about 1 case per 250,000 herpes infections per year. And so it's relatively rare and it's serious. It's 70% mortality if untreated. And in humans, we see 2 peaks of incidence of serious encephalitis. There's one at between 6 and 3 months of age. 6 months and 3 years of age. And that's when kids get their primary herpes simplex virus infection. So in a subset of those kids, the virus gets into the CNS. And then over 50 years of age where it's involving reactivation. So these older individuals are latently infected, and reactivation, again, most of the time goes to the mucosal surface, but very rarely goes to the CNS. So the question is, why does this happen? Why do these young children and older individuals get rare herpes simplex encephalitis? So what was done was to collect a number of individuals who had experienced this herpes encephalitis and who had recovered, and to do their genome sequence. This is called genome-wide association study, where you sequence the genome of an individual or a collection of individuals with serious viral disease, and you compare them to individuals who have had herpes infections but didn't experience encephalitis. And you hope to identify single nucleotide polymorphisms, changes in genes that are associated with that virulence phenotype. We call them SNPs because in a population of humans, the genome sequence everywhere varies among individuals in the population, and you can get many different sequences at any particular codon. So they're called single nucleotide polymorphisms that can lead to amino acid changes. And for herpes simplex encephalitis, a number of genes have been implicated, but here are just 4 mutations in TLR3, UNC93B, TRIF or TRAF predispose human carriers to herpes simplex encephalitis. So by sequencing the genomes of people with this condition, you can find mutations in the genes encoding these proteins that you can then introduce those into cell culture and actually prove that they have a role in infection. And TLR3, of course, is a sensor of virus infection, and it sends signals through this protein TRIF, and then downstream of TRIF is another protein, TRAF3. So apparently the TLR3 signaling pathway is essential for keeping herpes simplex virus's replication low in the infected individual so it doesn't invade the CNS. But if you have mutations in the genes encoding these proteins, now you have higher virus replication, more likelihood to invade the CNS. UNC93B is an ER protein that's involved in the proper assembly of TLR3 in the endosome. So again, defects in that protein lead to lower TLR3 and the same effect. So that's one example. And many, many different diseases are being addressed in this way because for every virus disease, there's always a subset of people with mild disease and a subset of people with serious disease. And we'd like to know the genetic basis for that. And currently we know for SARS-CoV-2 and COVID-19, some individuals get very mild disease or asymptomatic infections, and others get very serious disease. Can we use GWAS to identify mutations in cellular genes that are associated with that phenotype? All right, one more example of this, and this has to do with a child. One child is studied here who had severe respiratory tract infections throughout the first 5 years of life. And this is a study to understand why the child had so many rhinovirus infections. So in panel A here, we have on the x-axis years, so the first 5 years of life of this child, and all the different viral infections sustained over those years. And you can see plus stranded RNA viruses, a lot of rhinovirus infections. Each triangle is a separate episode with rhinovirus. And then the child had a number of coronavirus infections. These are the endemic coronaviruses that cause common colds. Influenza viruses, respiratory syncytial virus, parainfluenza virus, adenovirus, a DNA virus, of course, and then some bacterial infections. So this child was particularly susceptible to multiple and severe infections. So they sequenced the genome of this child and sequenced siblings and parents of the child as well. So if you can get all of that information, then you can look at the inheritance of any SNP that you identify. And so on the right are the sequencing traces in the MDA5 gene. It turns out that MDA5 mutation is associated with severe rhinovirus infection. So we have the father, the mother, the sister, brother. The proband is the patient. And they all have the proband and some of the other individuals in this family have a single base change in the MDA5 gene. So here's the whole MDA5 gene here with the different exons that are part of the protein, C-terminal domain, the card domain, the helicases. And you can see there's a single amino acid change here from lysine to glutamic acid, that amino acid 365 of the protein. So the proband, the child in this study, is homozygous for this mutation. And cell culture studies show that this single amino acid change abrogates the function of MDA5. If you recall, MDA5 is a cytosolic sensor of virus infection. It senses the presence of RNA and then turns on the interferon pathway, the secretion of cytokines, which of course are all important for resolving infection. And this child can't do that. So it gets a rhinovirus infection, cannot control it very well, and it gets multiple rhinovirus infections. You can see the inheritance of this mutation. Both parents are heterozygous. And so, one outcome is to have a child who's homozygous. And of course, one of the siblings is wild type, which you would expect, and one of the siblings is heterozygous. And so this child, the proband, had serious infections throughout its lifetime. This kind of analysis can be done, as I said, for a lot of viruses. If genomes are sequenced at birth, eventually we'll have a catalog of SNPs that are associated with severe, not only viral, but bacterial and other diseases, and we can consider interventions at an early year, if so indicated. Another host gene involved in viral disease is here's one that controls susceptibility. That is the ability of a virus to get into a cell. And this is in one of the receptors for HIV. And this is a figure we saw a while ago. At the top is HIV glycoprotein binding to its two receptors, CD4 and CCR, one of the CCRs. It can be CCR5 or CXCR4. In this case, the gene is the CCR5 gene that we're looking at. Both are needed for virus entry. And there are some individuals who have a mutation in the gene encoding CCR5. It's called CCR5 delta 32, and those individuals are protected from HIV-1 infection. This deletion is present in 4 to 16% of individuals of European descent. We don't know why it's there and why it's been maintained. What was the original selection force? There have been some hypotheses, but I don't think any of them have panned out. This mutation has been used multiple times now to cure individuals of AIDS, of HIV-1 infection. The first, a number of years ago, was called the German AIDS patient, and you can find out exactly who it is by Googling this because it's all public. This individual had not only a leukemia, but had AIDS, and he was going to receive a stem cell transplant to cure the leukemia. And his astute physician decided to find a donor. It was going to be an allogeneic transplant. To find a donor that had the delta 32 mutation. He found one. The German individual was given the transplant. They destroy the bone marrow. They replace it with the transplant. The bone marrow populates the individual, and then all the immune cells are produced from it. So the new T lymphocytes, the CD4 positive T lymphocytes, which are infected by HIV, now have no CCR5. They can't be infected. So he was cured, and subsequently another patient from London was cured as well. This is not a good way to cure HIV infections because stem cell transplants are risky and very expensive, but it shows that this gene is a determinant of susceptibility in people. Other determinants of virulence include age. Typically, very young and very old humans are the most susceptible to disease. This is exemplified in this graph of influenza in the U.S. between 1911 and 1915. We have death rate on the y-axis in age group, and you can see very high mortality in 1 to 5-year-olds, much lower in between, and then above 65, high mortality. Why is that? Well, we think the young have an immature immune response, so they can't combat infection. But obviously this isn't true for all infections, because we see with the current COVID-19 disease that is very rare in young kids. Older individuals being more susceptible, this is very typical, not just for influenza, but COVID-19. We think some of the contributions, they have less elastic alveoli, weaker respiratory muscle, diminished cough reflex, a reduced rate of production of new immune cells. The bone marrow diminishes with age. Even at my age, I'm beginning to have much less bone marrow. And so you can't make new immune cells as they're needed. And so that all may contribute. And this can be studied in mice, in which age is a determinant of virulence. Some other determinants. In general, men have slightly more susceptibility to viral infections than women. Not always, but sometimes. And this is being studied a lot in influenza virus. We think that females have generally elevated humoral immunity compared with males, and it seems to be phylogenetically conserved among different species, not just humans, which suggests that it may be important for reproductive success. Here's an experiment, the results of an experiment where immune response to influenza vaccine was studied in adult males and females and older males and females. So they gave these individuals influenza vaccine and measured neutralizing antibody in the serum, and this is shown as the seroconversion rate in the population. You can see that the females seroconverted at a higher rate than males, 14 out of 20 versus 29 out of 30. However, that difference in older individuals goes away, and in fact, females have a slightly lower response. So again, this may be in younger females to help with reproductive success. These antibody responses, the enhanced responses in females, correlate with elevated hormone estradiol, which probably influences immune response in some ways. However, I do want to point out that pregnant women can be far more susceptible to lethal disease, and that's certainly the case with hepatitis A, B, and E in influenza. Other determinants where there's far less understanding of the mechanisms, malnutrition certainly increases susceptibility because our physical barriers and immune responses are compromised when you don't have enough food. And this is why measles is 300 times more lethal in developing countries than in Europe and North America. Cigarette smoking clearly increases susceptibility to respiratory infections, and more recently, perhaps vaping as well. And air pollution increases respiratory disease. In cities with different levels of air pollution, that correlates with an increase in respiratory disease. And this all involves most likely damage to the respiratory tract, the smoking, the vaping, the air pollution, just damage to the respiratory tract, and a damage to immune responses. Our next question, which statement about determinants of viral virulence is wrong? A, viral virulence genes can encode viral proteins. B, virulence genes can encode cellular proteins. C, they are the same in all viruses. D, they can be found in untranslated regions. E, they may encode immune modulators, which is incorrect. The answer is C. They're the same in all viruses. They are not. Every virus has a different set of viral virulence genes. The outcome of infection, the production of disease, has, as we said at the top, two components. And one component is what the virus does to the cell. And so let's very briefly consider how viruses injure cells. As you know, as viruses reproduce in cells, viral proteins accumulate. The cell responds programs of cell death, including apoptosis, necrosis, and pyroptosis. So this kills the cell. And so that results in what we call cytopathic effects. Cytolytic viruses, viruses that kill cells, induce cytopathic effects, not just inclusions in cells and effects on the nuclei and so forth, but also overt cell killing by induction of cellular pathways. Many viral genomes encode viral porins, which are proteins that form channels in membranes and allow cytosolic contents to leak out. That further will destroy the cell. Many viral gene products inhibit host protein and RNA synthesis. This leads to membranes falling apart, leakage of enzymes from lysosomes, cytoplasmic degradation, and finally, enveloped viruses induce the formation of syncytia. So in cell culture, you can see the clear progression of cell killing by cytopathic viruses, the production of syncytia, and clearly these effects in a host contribute to damage in various organs. So poliovirus replication in neurons destroys the neuron, and that will cause the paralysis. But pathology of an infection, the disease has a huge immune component. And that's what I want to talk about for the rest of today. This is called immunopathology, or too much of a good thing. The immune response can help you, of course, but it can also cause cell damage. In fact, the clinical signs and symptoms of disease, which include fever, tissue damage, aches, pains, nausea, are in many cases mainly a consequence of host responses. All of this can be caused by immune responses, of course, tissue damage by virus replication as well, but as I'll show you, in the case of non-cytopathic viruses, which do not kill cells, disease is usually a consequence of the immune response. So let's look at some examples of this. And here's a table summarizing different kinds of immunopathology, not only caused by adaptive responses here at the top, but also overexuberant innate immune responses. Very early in infection, before the adaptive kicks in, can make too many cytokines or chemokines. It's not regulated properly, and that can cause tissue damage, as we'll see. So we have T-cell mediated immunopathology involving CD8 positive T cells and different kinds of CD4 cells, Th1 and 2 shown here, and these are the viruses for which we know that this contributes. There can also be B-cell mediated immunopathology, which is pathology caused by antibodies. And there's a coronavirus where that's shown, a coronavirus of mice. So let's go over some examples of these to illustrate. Here's an example of a viral disease mediated by CD8 positive cytotoxic T lymphocytes. And this is our friend LCMV, lymphocytic choriomeningitis virus, which is a mouse virus. If you take a mouse and inject LCMV intracerebrally, and remember, you only need one PFU to do this, in 8 days the mice are dead. If you immune suppress the mice survive and they have a persistent infection. The virus is replicating but not causing any disease. If you then give these mice CD8 positive T cells from another mouse that had been infected with the virus, the mice die. So the virus is not killing mice. It's actually the CD8 CTLS that are killing mice. So what are they doing? Here's some experiments to show the damage caused by CD8 positive lymphocytes. Here on the top are some experiments done with LCMV. And we're looking at two different kinds of mice here, wild-type mice and mice lacking the perforin gene. Remember, perforin is one of the proteins of cytotoxic T lymphocytes that pokes holes in the membranes of target cells. So wild-type mice, when infected with LCMV, the dark green lines within 10 days are all dead. Perforin knockouts all survive. This implicates cytotoxic T lymphocytes, CD8 positive lymphocytes, and particularly the cell killing mediated by perforin. And as a measure, another measure of virulence, instead of the death rate, we can look at the production of a liver enzyme, GLDH, in serum. And normally this enzyme is confined to liver cells. So it's present in the blood as a sign of liver damage. And you can see in the wild-type mice, this liver enzyme becomes elevated after infection, whereas in the perforin knockout, it remains at baseline levels. So the perforin activity of CD8 positive lymphocytes is responsible for cell killing, which is part of what CTLs do. They lyse virus-infected cells, but that also causes, in this model, death and immunopathology. At the bottom is another virus infection. This is Coxsackie virus infection of mice, which typically causes infection of the heart and destruction of heart tissue. And in A, these are wild-type mice. And this is stained with a dye that shows you tissue damage caused by infection, so extensive tissue damage of the heart cells. And B, these are perforin knockout mice. You can see there's much less tissue damage. So again, the CTL response is very damaging. In many virus infections, it causes damage. When you get influenza, your chest hurts. That's because cells in the trachea are damaged by CTLs, and eventually they have to recover. There are also lesions associated with CD4 positive lymphocytes, the helper cells, the helper T cells. These make a lot of cytokines, and they recruit many effector cells that can cause tissue damage. These typically are neutrophils and mononuclear cells that can harm tissues. Their aim is, of course, to resolve infection, but they cause tissue damage. And typically the immunopathology is caused by release of proteases from these cells, reactive radicals, and cytokines that damage the cells. Let's look at an example of this in humans. Herpes stromal keratitis, one of the most common causes of blindness in developed countries. It's almost entirely immunopathological, and it's caused by CD4 positive Th1 cells. What happens here is that the virus is reproducing in the eye, and repeated infections typically cause opacity and reduced vision. So one or two will not do this, but as you accumulate infections over time, the cornea becomes opaque and leads to blindness. How does this work? The virus replicates in the corneal epithelium. So here's a section of the cornea. There's the corneal epithelium on the top, staining dark color with the nuclei very clear. And on the bottom is the stromal cells of the cornea. The virus replicates on the top in the epithelium, causes inflammation, and the cytokines, of course, diffuse to the underlying stroma. And these cytokines produced by infected cells, they recruit CD4 positive Th1 cells, and you can see in this second section, which is the same section of cornea, just a lower magnification. There's the corneal epithelium, and then the stroma. Lots of CD4 positive Th cells attracted by infection in the epithelium, and these are producing cytokines themselves. They recruit neutrophils, and that causes swelling and damage of corneal cells. Blindness is not a direct consequence of viral destruction of the epithelium. It is immunopathological. Another example, West Nile virus in about 5% of infected individuals can develop encephalitis, an infection of the brain. In mice lacking toll-like receptor 3 gene are more resistant to lethal infection because they have impaired cytokine production. The observation here is that TLR3 mice are more resistant to encephalitis. Remember that TLR3, toll-like receptor 3, is a sensor of viral RNA. When it senses virus infection in the periphery of infected animals, that leads to production of cytokines, and included is TNF-alpha, which breaks down the blood-brain barrier and lets virus get into the brain and cause encephalitis. Another example of immunopathology. And here is an infection in mice to illustrate this. So we're looking at the brain of mice. It's been removed from the infected mice, and these were mice infected with wild-type West Nile virus. These are wild-type mice infected with West Nile virus. And the mice, after infection, are injected with a dye, a blue dye that will get into the CNS if it's highly permeable. So you can see day 3 after infection of wild-type mice, the dye is infused throughout the brain, and that's because of the production of TNF-alpha in response to TLR3 signaling. In mice that lack the gene for TLR3, you can see on day 3 there's far less dye in the brain, and not until day 5 do you have a small amount. So it's the TNF produced by TLR3 signaling that allows, that compromises the blood-brain barrier and lets the virus in. So it's immunopathology because it's a consequence of the immune response. Poxes and rashes associated with virus infection, measles, smallpox, varicella zoster. There's a child with measles rash. These are typically immunopathology reactions. And what happens is that There are infected foci in the skin. The virus has reached the skin after a secondary viremia. Th1 cells and macrophages home in on these foci. They produce cytokines, and the cytokines increase capillary permeability. They cause an influx of T cells, and that's what each of those little rashes is, caused by these cytokines. It's an immune reaction. Let's talk about an example of immunopathology caused by antibodies. And the example here is dengue fever or breakbone fever. This is caused by dengue virus, a flavivirus transmitted by Aedes aegypti mosquitoes. It is endemic in areas where these mosquitoes are numerous in the Caribbean, Central and South America, Africa, and Southeast Asia. Billions of people are at risk for infection. And we see at least 400 million infections a year. It's second only to malaria among insect-borne diseases. And I should say, as temperature of the globe rises and the range of these mosquitoes increases, so too do the number of individuals who are at risk for infection with dengue virus. At the top is a map of the world showing you the areas where dengue virus infections occur, which typically coincide with areas where the mosquito is found. So the red dots are outbreaks, and many of these are likely imported cases in the northern central U.S. where there's very little Aedes aegypti. They're probably travel related, but you can see in these countries that are red, lots of Aedes aegypti there throughout Africa and Southeast Asia. Before 1981, the use of DDT had eliminated mosquitoes for much of South America, and as a consequence, there was very little dengue. So this is confirmed dengue prior to 1981. Of course, then we recognized that DDT had deleterious environmental effects, so its use was banned. The mosquitoes moved back, and then dengue returned, as you can see from 1981 to 2003, high amounts of dengue in these countries. The movement of these mosquitoes back to these regions in part was due to the used tire trade, and these tires are carried on transport vessels. They have water in them, which can't be removed, and the mosquitoes breed in the water, and they're transported around the world this way. The virus infection causes a disease called dengue fever. It's an acute febrile illness with severe headache, back and limb pain, and rash. The severe aches and pains in the bones lead to it being called breakbone fever. This is typically self-limiting infection. You recover in a week or so, but in 1 in 14,000 of these infections, you get dengue hemorrhagic fever, which is a life-threatening disease where internal bleeding can lead to a fatal shock syndrome. So there are 4 different serotypes of the virus, and there's no cross-protection. So if you're infected with dengue 1, you make antibodies, you have a memory, and then when you get, say, infected with dengue 2, you make antibodies to dengue 1, but they do not neutralize dengue 2. So the protection is specific. And if you have a secondary dengue infection, so in other words, let's say you have dengue 1, you have this 14-day illness, you recover, and then you get infected with dengue 2 or 3 or 4, then the incidence of hemorrhagic fever and shock goes way up from 1 in 14,000 to 1 in 90 for the fever and 1 in 50 for the shock. So this is an example of B-cell mediated immunopathology, antibody-mediated immunopathology. What we think happens is that at the primary infection, you have the generation of antibodies against dengue virus, it's serotype 1, say. Now you have a secondary infection. You have a memory response to the original serotype, so secondary with a different serotype. Those antibodies will attach to the virus particles, but they will not neutralize their infectivity. However, they can lead to infection of monocytes because the antibodies will bind to Fc receptors on the monocyte. The virus will now get into monocytes. So you will make more virus. The monocytes will release cytokines and chemokines, and that gives you the enhanced disease. In the original infection, the viruses typically did not infect monocytes because the antibodies are made late in infection, and they neutralize virus infectivity. The problem is the secondary infection where the viruses are not neutralized by the antibody. Rather, the antibodies lead to uptake into these cells, and cause more serious disease. So that's an example of a immunopathology caused by antibody. We think that immunopathology is also important in the disease caused by SARS coronaviruses. It's well studied for SARS-CoV, the original virus seen in 2002 and 2003, and it's likely to be also a problem or a part of the pathology with SARS-CoV-2. And these infections, SARS-CoV-1 and 2, are both associated with fever, cough, pneumonia, acute lung injury, and what we call acute respiratory distress, the inability to breathe, which requires providing oxygen. Certainly part of these pathologies are due to virus destruction of infected cells, but we also believe there is a contribution of a dysregulated inflammatory cytokine response called the cytokine storm. You basically make too much cytokines and chemokines. Normally the production of these is regulated, which brings down their levels after infection, but for some reason in SARS-CoV and perhaps SARS-CoV-2, the cytokine production is not regulated and they cause damage. And that's illustrated on the right here, where we have the two consequences of responses to virus infection. So we have a normal outcome where we have a protective and regulated inflammation. We have inflammatory response to a virus infection. You have reduced virus reproduction. You have an interferon response. You have infiltration of cells that protect against infection, that resolve infection. And then you have pro-inflammatory cytokines and chemokines, which eventually are regulated by the anti-inflammatory versions. This gives you minimal epithelial and endothelial cell apoptosis, reduced vascular leakage, optimal cellular and antibody responses, and effective viral clearance, so you have immunity and host survival. In the cases where we have pathogenic or dysregulated inflammation, as caused by a cytokine storm, we have high virus reproduction, which may be key, and these coronaviruses replicate very well in the respiratory tract. We have a delayed interferon response, which may be a consequence of viral proteins that antagonize the response. We have inflammatory monocyte macrophage and neutrophil infiltration, the production of very high levels of pro-inflammatory cytokines and chemokines, and very high infiltration. See, the four arrows here represent much higher levels of these two components than in the normal situation. And as a consequence of the enhanced infiltration and cytokine and chemokine production, we have enhanced apoptosis, increased vascular leakage, suboptimal T-cell and antibody responses, impaired virus clearance, and that leads to acute lung injury, acute respiratory distress, and death. And this is in part why antibodies to IL-6, a pro-inflammatory cytokine, are being tested to treat SARS-CoV-2 infection because they're part of the overproduction of pro-inflammatory cytokines and chemokines. The last topic I want to talk about is another effect of virus infection, which is immunosuppression. And this is a global reduction of the immune response caused by virus infection. This can happen if the virus replicates in cells of the immune system, if replication perturbs cytokine production and signaling, or by viral proteins that are viroceptors or virokines, as we talked about earlier, immune modulators interfering with the immune response, not just to the infecting virus, but to other viruses as well, or other bacteria even. Here's an example of immunosuppression during measles virus infection. Measles virus is a very well-known immunosuppressor. So here we have a child with measles, and the assay for immunosuppression is a tuberculin test, a TB test, where a small amount of tuberculin protein is injected just beneath the skin, intradermally. And if this individual has tuberculosis, you will have an influx of T cells, which are primed by the bacterial infection, into this injected area, and you have a swelling or induration that you can measure. This is a good way to tell if someone has been infected with tuberculosis bacteria. But here we're using it as an indicator of immunosuppression. So if measles is immunosuppressing, we should see a reduced induration here. And so here's the control for children who don't have measles, and you get 7 millimeters of induration here. And then we have children measured at the peak of rash, and you see there is absolutely no T-cell response. And that increases weeks after the rash. So as the child recovers from the infection, the T-cell response begins to recover as well. So a very nice illustration of how infection suppresses T-cell responses, measles infection. And this is one reason why measles virus infection can be so serious, because it causes this immunosuppression, which can lead to other lethal infections. How does it work? Well, remember, our antigen-presenting cells have recognized viruses or viral proteins or nucleic acids. They produce cytokines that then influence how the helper cells are going to differentiate into, helper cells that then help production of cytotoxic T lymphocytes, or helper cells that lead to the production of antibody from B cells. So in this case, this infected cell is producing IL-12, which causes differentiation of helper CD4 cells to the Th1 response, which generates the production of CD8 CTLS. Now, measles virus inhibits the production of IL-12. So you don't have the production of CD8s, which are needed for immune responses. Instead, you have a skewing to antibody production, which is not useful in resolving these infections. And so that's a form of immunosuppression. And so here, these T cells are absent in the children with measles, as we saw from the TB test. Instead, there's a skewing toward antibody production, and for viruses and other pathogens that require CTLs for clearance, and that's many of them, this skewing to antibody response doesn't help. Here are some 3 viruses that cause immunosuppression. We talked about measles virus, which infects monocytes, dendritic cells, and causes a skewing of the CD4 helper differentiation, and it leads to reduced T cells and enhanced infections. Rubella virus infects lymphoid cells, and that leads to persistent rubella infections. And of course, HIV-1 infects CD-positive T cells and monocytes, and that leads to AIDS, which includes opportunistic infections and cancers. So immunosuppression is an outcome of infection with a number of viruses and can cause more serious outcomes. In the case of HIV, if there were no secondary infections, it would not be a lethal infection. But in fact, CD4 positive T cell numbers go way down and you can't survive other infections. So now we have a better understanding of how viruses cause disease, and we're going to start by looking at examples of viruses that cause different patterns of infections. And the first is what we call an acute infection, which we'll talk about next time.